How does the process of meiosis now explain Mendel's two major principles? Let's first talk about the law of segregation. Okay, the fact that the different alleles actually get divided from each other. Okay, if we think about the alleles in terms of meiosis, and think about the chromosomes that they're on. So I'm going to give you a little diagram here to imagine our chromosomes with their alleles. So I'm going to do some chromosomes here, I think, with some thick marker. These are going to be the chromosomes from the mother. And these will be the chromosomes from the father. And we're going to follow our alleles. Okay, here's alleles on each one. Okay, these are actually the genes. Okay, I should back up. The places where I drew these lines are the genes, or the spots on the chromosomes where we find those alleles. And just for sorting's sake, I'm going to call these alleles big A, and big A, and big B, and little b. Okay? When these alleles go through meiosis, remember that first in meiosis 1, the homologous pairs line up across from each other. So we're imagining these two sets of chromosomes are homologous pairs. Here's our imaginary metaphase plate, or the middle line of the cell. They've lined up across from each other. Okay, so our alleles haven't been separated from each other yet. Right? The big A is still with the big A, the big B is still with the little b. But when we go through meiosis 2, and we line up those sister chromatids, this part of the cell gets divided in half and splits the sister chromatids, and this part of the cell gets divided in half and splits the sister chromatids. And if you remember, when I went over this with the meiosis vodcast, I said that we've got one cell here, one cell here, one cell here, and one cell here to make 4 gametes. Okay, hopefully you remember that. Each of those gametes now has one allele in it. So where did our alleles get separated then? It happened back in meiosis 2, when I put that dotted line between them that separated those sister chromatids from each other. That separated the alleles, or the big B got separated from the little b, and the big A got separated from the other big A. Okay, that's our law of segregation. So the law of segregation occurs in meiosis 2. So again, I want to emphasize that Mendel didn't know anything about meiosis, but he figured out based on his data that those alleles had to separate at some point when the gametes were being formed. It was later, when we learned about meiosis, that scientists realized that the second part of meiosis, that second division, explains that law of segregation. The second one, the law of independent assortment, that you're randomly going to get different alleles in different cells. This time we're going to start at the end of meiosis with our 4 cells. So there's our 4 egg cells or 4 sperm cells, whatever we've made. Here are some chromosomes inside them. We're going to put 2 in each cell. Okay? And on those chromosomes, I'm just going to put different colors so we can keep track of what is what. So this was a homologous pair with, let's say, green and yellow, or green and yellow, yeah, are going to go together. So there was a homologous pair. Make those the other half of that homologous pair. And we'll put pink and red together. Here was another homologous pair. Oops, this guy is supposed to be yellow. There's a yellow one. Okay. So, get my pink. Those colors kind of match, so hopefully you can remember those are a homologous pair. So we ended meiosis in this case with yellow and red and green and pink. Now, would it have been possible for yellow to end up with pink and red to end up with green? Well, the law of independent assortment says it has to be. It has to be random which chromosomes, and therefore which genes, and then again which alleles end up with each other. So let's go back and see why that is. Why could we have different mixing of these colors of chromosomes in the end. Well, you remember when you did your paper meiosis, I said that it's completely random how the chromosomes line up in meiosis 1. So if we go back and think about meiosis 1, got homologous pairs of chromosomes. There's my yellow X chromosome and my green chromosome. And then here's my pink chromosome and my red chromosome. Okay, this is meiosis 1. They're in their cell, and they split apart at the homologous chromosomes. These go this way, these go this way, and then they do one further division, so that in the end we've got 4 chromosomes, or 4 cells, sorry, 4 gametes. And the chromosomes that are in those gametes are dependent on which side they lined up on. So in this case, we'd have red in both of these, with a green in both of these. And on this side, we'd have yellow and pink. So now we've lined them up in a way that they ended up being red with green, yellow with pink. So yes, we could line them up to have that combination. Well, how were they lined up in the first place to get that other combination? Let's look at it again. If we put the yellow, oops, that's not the color I wanted. Put the yellow again across from the green. Has to be that way because those are the homologous chromosomes. But now this time, put the pink on this side and the red under the yellow. Now when they divide up, we've got yellow and red together, and they will divide into their 2 cells, and green and pink together, and they'll divide into their 2 cells. So the combinations of the chromosomes, again, are random depending on how they line up right here in meiosis 1. Okay, depending on which side of the equator they line up on. Now remember, you don't have just 4 chromosomes in your cells, right? You don't have 2 pairs. You have 23 pairs for a total of 46 chromosomes. So imagine all the different combinations of ways that they could line up along that equator that would give all the different types of diversity to the gametes that you produce. So that law of independent assortment is where we get a lot of the differences in gametes that are made. So the reason, in other words, why you do not look identical to your brothers or sisters. Okay, the lining up of the chromosomes is different for each egg or sperm cell that's made.